In the last video, we talked about how you could take DNA and copy everything in the DNA, both sides, both strands, and then replicate a cell. And the topic of cell divisions coming up later, but that was called DNA replication, where you copied everything in copied all the DNA, and you would use that when you're trying to make an exact replica of a particular cell. What we're going to talk about now is how you can take a cell and instead of copying all the information in it, you're only going to copy a piece, a little section of it, and you're going to use that section then to make a particular protein. And that is broken down into two processes called transcription and translation. And together, transcription and translation result in what they call protein synthesis. So what's going to happen is you have your DNA and you're going to copy a piece of DNA, and it's going to start in one section and end in a particular spot. And that section starting in one spot and ending in another is going to be what we call a gene. So a gene is going to be a particular section of DNA that ultimately is copied and used to make a protein, as you see down here. So you're going to copy that DNA, and you're going to only copy a piece of it, and eventually that piece is going to be made out of a slightly different molecule than DNA, and it's going to be called RNA. And this particular piece we're going to call mRNA, and it's going to be the messenger RNA. This all takes place inside the nucleus of a cell, and then that mRNA is going to come out and it's going to travel along and it's going to be hooking up with ribosomes, and then the ribosomes are going to act as a meeting spot for the mRNA and this other kind of RNA called tRNA. The T stands for transfer RNA, and the transfer RNAs are going to match up with the mRNA, and on one side you're going to have a matching code that is the opposite of what you see on the mRNA. So they're going to match kind of like they did in the DNA with base pairing. And then what you're going to also have is you're going to have these amino acids on the other side of the tRNA, and that's how you're ultimately going to read it. And that process down there is called translation. Just quickly to mention some differences between DNA and RNA. In DNA, DNA is normally double-stranded. There are a couple of exceptions if you're talking about viruses and things like that. But for what we do in this class, the DNA is going to be double-stranded, RNA is going to be single-stranded. Remember, there's a different sugar in DNA. You have deoxyribose as your sugar. In RNA, you have ribose. There's that extra oxygen there. And then the other difference is with the bases, the nitrogenous bases. They're all the same except in DNA, you're going to see thymine. And in RNA, you're going to see uracil. So wherever you have adenine on one side, what's going to pair with that is going to be uracil. Whereas in DNA, if you have adenine on one side, what would pair with that would be thymine. So that's a difference that you'll see also there. So, well, what you see here is very similar to what we had with DNA replication, where we used DNA polymerase, remember, to copy the DNA and hook nucleotides together. But instead, what's going to happen here is we're going to hook RNA nucleotides together. So this greenish colored round circle here represents a different enzyme, and it's called RNA polymerase. And what it does is it hooks RNA nucleotides together. So here you have your DNA, which is the red or pink color down here, and this shows you the particular sequence. And you'll notice that G pairs up with C, and G pairs up with C, and A, notice, pairs up with U. So anytime you see U, uracil, you know you're talking about RNA. So you're going to end up copying that section, and what happens then is it turns out there's a little bit of editing that often goes on. And so when you copy your mRNA, it turns out you'll have pieces of it. And some of the pieces some are called exons, some are called introns. And you'll notice here you have an intron and an intron and these 3 exons. And what happens is there are enzymes that will cut out the introns. You can think of that as the introns go in the trash. And so this whole process is called mRNA editing, or RNA editing, I should say. And you end up with when you hook your exons together, that sequence then becomes your mRNA. And that's what's going to then go out and be read by the ribosomes. Now, your DNA, comes in these individual nucleotides, G, C, A, and so forth. But what we're going to do when we have our mRNA, which is also individual nucleotides, we're going to read them in sections of 3. Okay, so a section of 3 in a row is going to be what we call a codon. And a codon is going to be used to represent a particular amino acid. So you notice that this codon here, CGU, is going to be the code that codes for arginine. And UCA is going to code for serine and tryptophan and so forth. Okay? This is a code sequence, this is a chart of what the mRNA sequence would be if you had your mRNA and what amino acid you would get. You don't need to memorize this chart, but you do want to make sure you understand how to read it, and you want to also make sure you understand the fact that this is an mRNA chart. So if you have a sequence of mRNA, you can use this chart to figure out what the mRNA would code for and what amino acid you would get. Okay? Now the next part is looking at how that all comes together. So here we have our mRNA, and you can see here's our ribosome, and what's going to happen is the mRNA is going to go through the ribosome and read it in pieces, and the molecule here is your tRNA, and that's going to match up with its anticodon with the codon you see down here. You'll see that in more detail in a second. So here in blue is the tRNA molecule, okay? And the tRNA molecule has a couple of important things on it. On one side, it has a particular amino acid sequence, okay? And then on the other side, it has what they call an anticodon, a section of 3 codons that are going to be the opposite or the anti of what the codon is on the mRNA. So if the codon is CGA here, the anticodon is going to be GCU in this case, because the G will base pair with the C will base pair with the G, and the U will base pair with the A. All right, so in this picture, once again, this shows you, here's your P site and your A site. I'll go over this again with a little more detail in a second, but this shows you how you're building your protein here. So your P site and your A site, and what happens is new things are coming into the A site, and then whatever's attached to the P site is going to move over. Okay, whoops, went too far. All right. There we go. All right, so what happens here is don't worry about this beginning part. We'll just jump down to here. What happens is that your ribosome actually comes in sort of pieces, and it gets built sort of as you start this process. But we'll just sort of assume that the ribosome is together, and we'll just learn it this way. It just makes it a little bit easier. So what happens is your ribosome has a couple of spots on it. It actually has a third spot over here, but in Bio 1 we typically don't put in this site over here where they exit, but that's okay. So we'll talk about these 2 sites, or these 2 spaces, the P site and the A site. So what happens is your tRNA molecules will come floating in and will base pair with their appropriate anticodons. So the anticodons, once again, the anticodons are on the tRNA and the codons are on the mRNA. So you have the P site and the A site. What comes in to the P site, and then something will come into the A site, and then what's going to happen is the amino acid that's attached to the tRNA on the P site is going to move over and attach to this one. So this amino acid here is going to move over and attach to this one. So these two will be connected. This one then is going to float away and exit. And then this whole thing, the tRNA and the mRNA connected, will shift down one. So what's in the A site, the whole thing, is going to move from A site to the P site. That will open up the A site again for your next sequence, for your next codon sequence. So you're going to do that in order until you reach the end. And once you reach the end, that final product, the stop codon, will end up being your amino acid sequence, and it'll be in that particular order. So once again, if you look up here in this picture, here's my polypeptide chain I'm building. Here is my P site, okay? And notice this amino acid is shifted over and connected right there. So what's in my P site now is just a tRNA that no longer has its amino acid, so that floats away. And then this whole unit here, the mRNA and the tRNA, the whole thing shifts down one. So what was in the A site all shifts down to the P site, as you see here, and then that opens up the next spot. And once you open the next spot in, the next tRNA can come in, and then you repeat the process once again till you get to the end. Just to show you how many different base pairs there are, genome size and base pairs of different things. Here's a human, for example. We have about 3 billion base pairs with 20 to 30,000 genes, and a chicken and a fruit fly and so forth. Notice that humans are up here pretty high, but not necessarily is there a link between how many base pairs you have and how complex the organism is, because you notice a pea plant down here has 5 billion. So we'll stop there with that.